Thanks very much, Jörn, for the kind introduction and also for giving the opportunity here for me to talk. I would like to follow some of the preceding speakers in expressing my joy to be back here in Ambleside. It's really fantastic, but also to be able to be at a real conference where we meet people. It's all just so much better, really. It's just irreplaceable, basically. So as Jörn was already saying, for the last few years, we have been working on G-protein coupled receptors. We're trying to understand the dynamic landscape of these receptors. However, my talk is in many ways much more qualitative comparing to the preceding speakers' talks, which really went into the nitty-gritty, doing quantitative analysis. Insofar, our investigations are really limited by many different aspects. For example, the ability to make samples, the stability of samples, concentrations, NMR sensitivity, etc., etc. And so we really bound to primarily relatively simple means of NMR spectroscopy. And for me, it certainly has been a lesson sort of stepping coming down many steps, sort of moving away from triple resonance experiments and suddenly finding ourselves in doing relatively simple experiments, looking at chemical shift changes, intensity changes, etc. And this is sort of what I would like to sort of convey in my talk, that even using relatively simple means, it's actually NMR is really powerful in what we can do. And of course, sort of our intention is, so G-protein coupled receptors is highly dominated by structural biology, conveying a static view of these receptors, but they are very dynamic, these proteins. So they're membrane embedded signaling protein. They're basically the sensors of the cell, and they monitor the appearance or the presence of a whole range of different ligands on the outside of the cell. They're very specialized, so they bind a particular set of ligands in the receptor we are investigating. In particular, this is the natural ligands adrenaline and noradrenaline, and they report the presence of these ligands through binding them, which then leads to conformational changes in these 7 transmembrane membrane helix receptors. And then that allows to bind on the cytoplasmic side of these receptors various signaling proteins. Now, as the name gives away, primarily these are heterotrimeric G proteins which are being bound, and then this leads to the propulsion of signaling cascades, but they're also binding a range of different proteins, for example, arrestins and many other binding partners. So crystallography in the last 20 years has made, and now increasingly cryo-EM in the last 20 years has made huge progress. There's now over 500 structures of over 100 receptors, and this has really shown us these receptors in various states, in particular inactive, so switched off, and active states. Crystallography in cryo-EM is only able to show these active states once they're bound to the binding partners on the cytoplasm. And thanks to these massive efforts, we really understand now that there is a whole range of motifs on these helices, which are sort of on the limit of stability, which relatively easily change some, say, hydrogen bonds or something like this, which then leads to changes in these helices and allows these receptors then to function in a dynamic way, changing conformations and then contribute to the signaling process. So as I said already, heavily dominated by static investigations. However, these receptors are very, very dynamic, and that basically leads to the conundrum that many pharmacological events are not really explainable through the static view. So for example, partial agonism, so there is ligands which are binding to these receptors which don't lead to the full response, so they only partially switch on, so that this is measured in efficacy, or also in terms of biased agonism, depending on if these receptors now signal via their heterotrimeric G proteins, or, for example, through beta-arrestin. And this is all at the moment very, very difficult to understand just based on a static picture. And this is where we intend to use NMR to really drill into these questions and to use NMR really in the integrative approach in a complementary fashion. And on the right-hand side, you can see the adrenergic receptor beta-1 in two states, apparently. So one's bound to an inactivating ligand, one's to a fully activating ligand, but due to crystallographic constraints, basically, they're almost superimposable. So sometimes crystallography also doesn't get it quite right because they have to use all sorts of different tricks. Now, the system we are working on is called beta-1 adrenergic receptor. We would love to work on the human receptor. However, this one is very unstable. So we're working on a turkey form of it. And even the turkey form, we're actually using a stabilized form. Now, there's a little bit of stabilization through 3 thermostabilizing mutations. So the properties of this receptor will be a little bit varied from wild type. But this is basically as good as we can do at the moment in our case and in our hands. So this is the receptor we're using. We're expressing it in insect cells. We're using methionine labeling. And as you can see on the right-hand side, on the structural sketch, the red dots basically show the methionines. They're reasonably spread across the receptor, and that in the worst case scenario at least gives us some relatively low resolution idea of how this receptor will be working. Sorry. Right. So activation, so the beta-1 adrenergic receptor basically increases heart rate. Increases the heart rate and also contractility of muscular cells. And the native ligands are adrenaline and noradrenaline, but because this is a pharmacologically very important receptor, of course, there's a wide range of different drugs available. Some of them are fully activating, others are inactivating, so-called, or reducing the activation, so-called beta blockers, and they are, of course, all competing against the native ligands. And so we can use these ligands now to start our investigations, and you can see here a range of spectra where each of the colored spectra is basically showing the receptor bound to a different drug. And we have got the assignments of these individual resonances through mutagenesis, so no triple resonance or anything here. And of course, it's very difficult to see what is going on here, but if you look at the bottom right spectrum, which is showing the receptor bound to a full agonist, then you will see that probably the spectrum looks a little bit more empty, so some signals are disappearing there. Now, if we use, of course, our favorite analysis software, we can easily overlay these spectra, and we see here already zoomed in into some of the regions, and you will see, for example, there in the middle left, methionine 223, which is on helix 5, or on the right, methionine 296, which is on helix 6. They seem to be gradually shifting in what sort of looks in a linear form, depending on the ligand which is bound, and the efficacy of the ligands is shown at the top of the page. So basically what it says, with increasing efficacy of the ligand which is bound, the signal is shifting more and more in the same direction. So, of course, as NMR spectroscopists, this will, and Chris has done already the work here, basically we know immediately that this is some form of two-state exchange happening on a fast time scale. Now, instead of looking at a titration where you're adding more and more ligand here, we can reproduce the same here by basically adding a ligand which has a higher efficacy, and that provides basically exactly the same result, that it's shifting this AB equilibrium more towards the right. So we can already propose here what is going on because we know the properties of the individual ligands we're binding, and so we have an exchange between an inactive and an active form of the receptor, and this exchange is happening fast on the chemical shift timescale. We didn't spend any particular effort to trying to exactly work out what the exchange rate is, but from the chemical shift difference, we can say already that the rate must be larger than 1,000 per second. Okay, But the story is a little bit more complicated, as it turns out. So when you look at that box in the middle, methionine 223, then you can see that there is some temperature dependence. And in fact, as we're lowering the temperature now of the spectrum, we're recording, then you can see that that broad signal is starting to split up in multiple peaks. And when we measure the intensities, they're plotted on the right-hand side, then you can see that basically the intensity is going up. So here we have an exchange situation which starts out on the other side of the coalescence temperature. So we're in this sort of intermediate to slow exchange regime. And as we're dropping the temperature, it slows down. So it favors the line shape, makes the signals become sharper. So it basically means that the picture we had developed previously was not quite complete. And it means that this active state is not just a single conformation, well-defined, but it is really a superposition of multiple conformations. We don't know how many, but it is continuously exchanging on a relatively slow time scale between these different conformations. So we imagine that maybe each of these conformations has a particular propensity to favorably then bind one of the cytoplasmic binding partners, and this might be the implementation of the receptor, how it can then select which one of these cytoplasmic binding partners it wants to interact with. Okay, so following that, we wanted to understand what exactly is happening after the ligand activation and once the receptor would be binding its cytoplasmic binding partners. Now, in an ideal world, this would be the heterotrimeric G-protein, but because all of the studies I'm showing you here are basically done in detergent micelles, it actually does turn out they are not particularly nice towards cytoplasmic proteins. They unfold, aggregate, so we need to use some kind of a surrogate. And crystallography uses nanobodies as surrogates because they can fix, they basically interact with the active conformation of the receptor and fix these GPCRs in the active conformation. So these nanobodies have no biological function at all. They are just here as a tool to trap the receptor in the active conformation. So we wanted to emulate this by NMR. So we're starting here with a receptor which is free of ligand, no ligand bound. And you can see on the bottom right, there's this little cartoon of the receptor which shows us all the methionines. So at the moment, they're black. So we're adding now our isoprenaline, which is a full agonist. So it fully activates the receptor. And you can see across the spectrum that there is various of these signals which are starting to move. Some are disappearing. And on the cartoon, on the right, you can see, for example, at the top 178, so this is something near the binding pocket, so not surprisingly, that shifts. So all the ones which have switched to orange, they're shifting. But you can also see now at the bottom of the receptors on the cytoplasmic side, there are changes happening as well. So these receptors are highly allosteric, so that basically conformational changes are happening at very long distances from each other. So the binding pocket is towards the top of the receptor, but this transmits now also to conformational changes on the other side, so on the cytoplasmic side of the receptor. Okay, so now we're stuck. Thanks. So now we're adding this nanobody. So we're forming what we call a ternary state. And now you can see that the conformational changes or the chemical shift changes are rather huge. They're really big. And now when you look at the cartoon on the right-hand side, you can see we have changes on the cytoplasm. Yes, makes sense, because this is where we're binding our nanobody. But now it also transmits to changes on the outside of the receptor. So there is this kind of allostery which goes one way towards the cytoplasm when you bind the ligand, but then you bind something on the cytoplasm and it transmits its allosteric to the outside. So we have these receptors which are constantly communicating between the different areas. It's still stuck. So basically we repeated exactly the same experiment as we've just done. We formed these ternary complexes, but we did them now for a whole range of different ligands, and these ligands just varied in their efficacy. And we did this from very low efficacy to very high efficacy. And what you can see there again is that basically some of these positions, some of these methionines are shifting in what looks like a reasonably linear fashion. It's not perfect because you need to imagine that the efficacy is measured in a cellular context, sometimes using constructs which might differ from the one we're using to do NMR. So this is not perfect, but I hope I can convince you that this looks reasonably linear. And linear to us, of course, means again that we have some fast exchange, but this time it's between two ternary states. And for the moment, we are just calling them a more active and a less active conformation. So we can corroborate this by lowering the temperature. We're looking at the carbon shift, and we can see that the carbon shift is changing upon reducing the temperature. So that means that basically the thermodynamics of sampling are changing and that the distribution between the two conformations is changing. Could you move? Thank you. So actually the carbon shift information is quite informative in terms of what is going on with the chi-3 rotamer position. So this is really the dominant influence. And so, for example, in the diagram on the left, you can see for methionine on helix 6, methionine 296, when we're in the inactive state, the side chain rotamer is in a trans position. Then when we add an activating ligand, it goes into this trans-gauche exchanging area. And then when we form the ternary complex, it then basically goes into a gauche conformation. So this is something we understand very well. So what we did now is we repeated these temperature variable experiments for a whole set of complexes. So ternary complexes in the top box and just ligand-bound receptors in the bottom box. And what you can see is that if we compare the chemical shifts for the ternary complexes, then the signals upon reducing the temperature, the signals are basically moving up. So that means they're going towards smaller ppm. And we know from the chemical shift information that this corresponds to a gauche conformation. So if we imagine that this is an entropy-driven process, then basically upon lowering the temperature, obviously it's going to adopt the conformation which is entropically more favorable. So from this we deduct therefore that the gauche conformation will be more rigid. Now if we look at the ligand complex, it goes towards larger ppm as we're reducing the temperature, and there it goes from a gauche trans sampling towards a more rigid trans conformation. Now, what that means, and we know that because of the titrations we have done with the different ligands, the ligands of different efficacy, we know that if these ternary complexes are moving now more towards the north, as we're reducing the temperature, so towards smaller ppm, and getting a more rigid conformation, then this means more rigid stands for higher efficacy. So a ternary complex which is more rigid, so which is in the correct conformation, is more active. With the ligands, it's pretty much the opposite. So upon reducing the temperature, it goes to the trans conformation. The trans conformation is more rigid, but corresponds to a lower efficacy state. So more rigid for the ligand-bound state means less efficacy. In the ternary, more rigid means better signaling property. Okay, in a model, we're speculating that this might be this fast conformational exchange between two ternary conformations. On the right-hand side, you see really the optimized conformation, which interacts ideally with the heterotrimeric G-protein and accordingly allows to release GDP in a very rapid way. On the right-hand side, you can see a form which is more dynamic, less well optimized for heterotrimeric G-protein interaction, and accordingly would release GDP slower. Now, of course, we haven't done any of these experiments with heterotrimeric G-proteins. This is all speculative, and all the information we have obtained comes, of course, from nanobodies. Right. So when we're looking at the signal on helix 5, 2 to 3, then you can see that this looks a little bit kind of dodgy as we're moving towards higher efficacy ligands. Basically, the intensity is dropping, so we can use the intensity of these signals sort of as a mimetic of how much microsecond, millisecond dynamics is present. So we call this an order parameter, but maybe this is not a very lucky kind of description. So it's just a measure, basically, of how much microsecond to millisecond dynamics there is. And so we have done this for a whole range of ligand-bound, apo, and ternary complexes. What you can see in blue there in the middle is basically the ligand-free. And on the x-axis, it shows you basically towards the right, increasing order, so less dynamics. And towards the top, it shows you helix 5 with more order, so less dynamics as well. So when we go from the apo towards the ligand-bound states, you can see that they're more dynamic. They show you smaller numbers, in particular for fully activating ligands like adrenaline and isoprenaline. And the ternary states are generally more ordered. They're the red ones on top. And tentatively, we can also say that there remains more motion on helix 6 than there tends to remain on helix 5. Okay, so the problem with our studies so far is that they give us a relatively limited view of what's going on in which part of the receptor. In particular, our problem was that we have this methionine on the cytoplasm of helix 6, M283. However, because this is extremely solvent exposed, it doesn't really pick up any conformational changes that are taking place. In addition, we don't have any probes on helix 7, and helix 7 contains one of these very important activation motifs, a so-called NPxXY. So we kind of had to switch technique, and we moved now from carbon NMR to 19F NMR. Of course, there's no fluorine in this receptor, and we have to attach probes, and we do this through attachment to the side chains of cysteines. So we have one native cysteine on helix 7, and we put in a mutant on helix 6 to attach also a probe there. So we look at either one on helix 7 or one on helix 6. There's a wide range of different probes available, and this is actually an active research field, but we just went for the one there on top in the middle, TET, which is relatively small and simple. Doesn't give the best chemical shift dispersion, but we really thought that introducing something which was as small as possible was kind of the key factor for us. So you can see this is a two-step reaction basically to attach that fluorine probe. It's attached by a disulfide bond, and then on the bottom right you can see we can record now the spectra in a very clean form. Either we see the probe on helix 6 in green, or we can see it on helix 7, and that gives us the blue spectrum. So one peak as we would maybe expect. And so we can investigate now helix 6, and we can see that for the different efficacy ligands, this leads to some variation. We can't quite convince ourselves that there is a linear dependence between efficacy and chemical shifts, but obviously there are some small changes happening. And when you can see what says there, NB6B9, this is basically when we form the ternary complex, there is a massive shift involved. So the ternary complex is substantially different from all the other ligand-bound states. If we do this on helix 7, and we look at all the different complexes in following increasing efficacy of the ligand-bound, then you can see that the main signal is gradually shifting to the right. You can see also that the signal intensity is dropping. So that means we have broadening of the line. And again, you see this very characteristic jump to the left when we form, so the black spectrum, when we form the ternary complex. And we'll get back to that, hopefully, towards the end of the talk, if I don't run out of time by then. So for helix 7, it looks quite different, and we have a linear correlation now between efficacy and the chemical shifts. So again, it seems to suggest that we have a fast exchange, and we just make this very simple. So for the moment, we say two states, so fast exchange between two conformations, and because the exchange seems to be relatively fast, as I will show in a minute, it has to be between two inactive forms. It can't involve an exchange between inactive and the active form, because the conformational change would be far too big, and this would really slow down the exchange rate accordingly. So, as I said, line broadening, so CPMG. CPMG is something we can do with fluorine NMR relatively easily, and where we miserably failed, for example, when we did carbon NMR, but Chris's talk was very inspiring, so we would like to go back to this again. So fluorine NMR, we can use 10 millisecond transverse period, and we can pulse, and on a cryoprobe, you can easily pulse up to 10 kilohertz or something like this. So certainly the high side is not a problem technically. It's the lower frequency, of course, which is always the problematic one, because that is very greedy on the transverse time. And we can do this at the lowest frequency of 100 hertz at the moment. And so we measured the the CPMG profiles at different fields for different ligands. And then we did a global fit, basically using Andy Baldwin's generalized equations, where we don't have to make any initial approximations if you're on the fast or on the slow exchange side of coalescence. And what we very nicely see here in the box on the right is, so we're in the thousands in terms of exchange. That's why I told you already, it has to be between two inactive forms. The exchange is very rapid. It must be to do with small conformational changes. We can see also that the exchange is accelerated when we go to more activating ligands. And we can also determine the population, and we can see that this activated form, the I2, is increasingly populated when we go to the more active ligands bound. So there were some MD simulations done on a related receptor, beta-2 adrenergic receptor in Ron Dro's group in the past, and they basically suggested that there is this tyrosine, which is just next door to our cysteine, where we have the probe attached, which just basically changes the phase conformation of the aromatic side chain. This is a relatively small change, but of course, in terms of ring current shift, very active, and therefore influences our proximal probe. So we suspect it's just a small flip of the aromatic residue, and this is basically what differentiates I1 and I2. So obviously this important residue, this important tyrosine, is moving in some pre-active kind of state. Okay, so this was the fast exchange. Now we scanned our spectrum using very weak saturation. So this is basically a CESS profile, what you can see here, and we realized that there is a response of the system. So when we look at the intensity ratio of the left-hand and the right-hand side of the peaks, that you can see this deviates slightly from 1, and it deviates more the more activating the bound ligand actually is. And it seems to be responding most at about 300 hertz towards higher fields. So we know something is there. We can now do this as a time series, so with increasing length of saturation, and so we can do the fits, and we get much slower exchange rates now, so below 10 hertz, and so we're concluding this must probably be now the conformational exchange, which switches from inactive to an active conformation. We can get the populations again. It increases the population of this active state when we have a higher activating, stronger activating ligand bound. However, it's still a relatively low population, and try to show this sort of graphically here. So we have this inactive fast equilibrium, then we have the slow inactive active equilibrium, and the active state is not as highly populated as we would think it should. And presumably, this is a consequence of the thermostabilization we're using with this particular construct. Okay, so ternary complex formation, again, through nanobody binding, we can investigate this now using fluorine NMR as well. So we looked at these ternary complexes now in the presence of different ligands. We see a nice correlation on the right-hand side between the amount of ternary complex formed and the efficacy. So we know the more ternary complex there is, the more efficacy, the more signaling propensity. The ternary complexes seem to be in the same shift position, so the conformation seems to be identical, and the signals remain sharp. So this is a picture for helix 6. If we do the same now for helix 7, then I'll remind you of what crystallography tells us. Again, is this tyrosine residue here, which is very important. This tyrosine does a very large conformational change once it switches into the active conformation, and it basically acts like some kind of a lock of the active state. So it protects the active state from switching back into the inactive form. So we did this now again, the various ligands in the ternary complex for helix 7. You can see that the efficacy plot still holds. So the more ternary complex formation we have, the more activating the ligand is. You see now the chemical shifts are changing, and they're also quite broad. So this is quite a different picture to what is happening on helix 6. Crystallography here tells us, so they have reproduced this as well now. So they have formed all these ternary complexes with nanobody with different ligands, but they all look totally identical in this region. And that really disagrees with our results, where we see basically our ternary complex in different positions at different chemical shifts and with different line widths. So of course we're scratching our head what this means. So we superimpose crystal structures of different complexes with different G proteins, etc., for different proteins, but they all seem to be looking exactly the same in this helix 7 region. However, if we superimposed two complexes, which were, one was formed with heterotrimeric G protein, and the other one with rhodopsin, then we can see, it's actually not very easy to see, but it's on the graph on the right-hand side there where that red box is, you can see that there are actually relatively subtle differences in the helix 7 region. So the difference obviously seems to be related somehow to either G protein binding or arrestin binding, and this is what's typically called biased agonism, because this is switching between signaling pathway. So we suspect that what we're seeing here by NMR, these changes, these subtle changes in conformation, are somehow related to biased agonism, and this seems to be corroborated in particular because ligands which are known as biased agonists, like carvedilol or bucindolol, they're all drugs, and they're all known to be biased agonists, basically seem to be pushing very strongly in this direction. Okay, so one thing which always has been on our mind is that the chemical shifts of these ternary complexes are so different. It's still less than a ppm, but this is a lot here. And that they're so different from just the ligand-bound states. And when we look at the structures, we don't really see any aromatic residues which are close enough, which would justify through a ring current shift changes that there would be such a large shift change involved. Scott Prosser's group has actually done some quite interesting work where they showed that the changes in solvent polarity really led to chemical shift changes, and that increasing polarity, which basically shift your fluorine signal towards the left, so towards lower field. So that would basically mean that the ternary complex would have easier access from water, so lower hydrophobicity, compared to just these ligand-bound states. So that's what the chemical shift analysis would tell you. And so a very easy way, of course, to corroborate this is by adding a paramagnetic label, gadolinium. This is called Magnevis, that one. And we're using this for a ternary complex where we actually can see both the ternary complex as well as just the ligand-bound form. And we're titrating now this gadolinium in, and the slope on the graph on the right-hand side basically tells you how much additional broadening comes from this gadolinium. And so the red line is steeper, and the red line corresponds to the ternary complex, and the blue line corresponds to the ligand-bound state. So that means it seems to be confirming what we insinuated from the chemical shift changes already, that the ternary state seems to be more solvent accessible, which is interesting because we would think obviously that docking this nanobody somehow must block solvent access, but here we go. This is what we can see. Right, I think this is really at the end. So this is the kind of all-inclusive multiple equilibrias going on. So depending on which part of the receptor we look at, we see different time scales, and we always have to be very careful that we don't generalize this on the whole receptor. So what, for example, the methionine NMR was telling us was really a region just below the binding pocket, so where the ligand binds, so between the ligand binding pocket and the cytoplasm. While with fluorine NMR, we really concentrated on probing the cytoplasmic side. And this is very, very important, and this also allows us to understand how this gear shifting in these receptors is happening and how the signal can be transmitted from one region to another one. So in a nutshell, basically a relatively simple experiment, and we would love to do much more complex things if we can. It is just that our hands are tied to a large extent, basically, to what the samples allow us to do. So the most important slide here at the end is a lot of the work I talked today was done by Nietlispach and Andres Sol during their PhDs, two excellent students, Andy Jones and Aditi Tandale, the current generation. Andy is here and gave a talk yesterday in the poster session. Mark Bostock helped with the data analysis, and so did Bill Broadhurst. Binesh at Novartis helped us with some expert advice in protein expression, and Chris and group basically really introduced us into the area, what is now many years ago. And BBSRC for funding, and thanks to you for listening.